So let's see what a pattern finding is. So a pattern or a keyword basically is an ordered sequence of symbols, and the symbols of the pattern and the search text they are chosen from some alphabets. So for example, if you are having the DNA, or alphabets are A, G, T, C. For the proteins, we have the alphabets which are 20 amino acids. So a pattern can be anything derived from these symbols. Same way, the search space or the sequence in which we are looking into the pattern is also derived from some alphabet. So there are 4 different ways in which we can find those patterns. We can look for a perfect match. For example, here we look for this pattern to be exactly same and its exact matches. So that is exact matching. We can allow errors due to substitutions. A substitution is where we have a mismatch. So in computational term, we call it as a mismatch. So if you look into this example, at position number 3 on the top part we have T, whereas in the bottom strand we have G. So in this way it's a mismatch, so we call it as a substitution. So maybe in nature there is a substitution, a mutation takes place where a nucleotide has been substituted. We can allow errors due to insertions and deletions. While putting them together we call them indels. So here is an example. So in the top strand we have a gap. In computational terms we call it a gap where we don't have a nucleotide over there, and on the bottom side we have some nucleotide which is C actually. So on one side in biological terms we can say there is an insertion. So indel might have 2 possibilities. On one side there might be insertion, on the other side may be deletion. So what happened in nature we don't know. If we believe that this C existed in the real world, then on the top strand there had been a deletion, and if it does not exist, then on the bottom strand we can say there is an insertion happened during the course of evolution. So this kind of matching is indels, insertion deletions. We can rank possible matches according to a weight function and keep the matches above a threshold. So here we can keep those matches that have some that are retained after over a threshold, so we can score them. So here what we do is we can have a pattern P1 and P2. There are 2 patterns of length 5. W is the weight of complete patterns defined via nucleotide weight function. So what if T replaces C? What will be the score? What if we get the exact match? What will be the score? So in the end we can score all these pairs. So where obviously where we have matches, exact matches, or scores are high. For mismatches we can have a scoring scheme where we can do some penalties. Same way we can treat gaps. So in this way in the end we come up with some score and we define a weight function. So for every pair aligned together we come up with this weight function, and in the end we combine all those weights together. So that is the overall weight for this alignment as we see in this example. So for all those 5 we are summing up their scores. We put them together and we will select if the score is above some threshold. So pattern finding in a genome can be presented in a generalized organism. So what is our expectation? So let's go into a little bit formal algorithms, and we say our goal is to find all occurrences of a pattern in a text. So our inputs are patterns. We have P1 through Pn, so that is the length of the pattern. So we have a pattern P and we have a text T. So T has a length T1 through Tm. The length of text is M. So these are our inputs to the system, and in the end what we get is we call it as output. So output is an indication that the pattern P exists in T or it does not exist in T. So here we have that elaboration in this diagram. So we have text. On the top we have indexes. So since we use arrays and we use those data structures, so we start those indexes from 0 and then it goes up to 13. So total the length of the text is 14 and the pattern is of the length 6. So pattern matches at position number 7. So what we will get in the end is that P is a substring of T, that P is equal to T starting from 7 to 12 position. So it's present within it. So in this section, what we have seen here is that pattern finding is an interesting job where we try to find some sequences within some larger sequences. And in bioinformatics terms, it is finding those patterns within the strings.